Hello, today we're going to talk about chapter 29, which is development and inheritance. Development is something that occurs throughout life. It starts at fertilization when the sperm fuses with the secondary oocyte, and it continues all the way until a human has reached maturity. We can divide this development up into prenatal development and postnatal development. And so that's what we're going to do. We're going to talk a lot about prenatal development. Prenatal development begins at fertilization or conception when that sperm fused with a secondary oocyte. And there are 3 phases to prenatal development. The first phase is pre-embryonic development. And this is the development in the first 2 weeks after fertilization. So as soon as the sperm and the secondary oocyte fuse together, and then through the first 2 weeks, there's a lot of cell divisions and differentiation where there's the formation of different types of cells, and this helps to create highly specialized structures. Then the second phase is embryonic development. And embryonic development will occur between the 3rd through the 8th week of gestation, so of prenatal development. And then finally we have fetal development, and that occurs from the 9th week of gestation or the 9th week of prenatal development up until birth. Postnatal development then is the development that occurs from the time of birth up until maturity of the human being. In the next video, we'll talk about inheritance. Inheritance or heredity is the transfer of genetically determined characteristics from one generation to the next generation. So let's start by looking at fertilization. So first of all, the maximum amount of time that a sperm can live in a woman's reproductive system is 5 days. Some sperm will live for a shorter time, depending on the conditions in the vagina, the fallopian tubes, and the uterus. For example, if the consistency of the cervical mucus from the female's cervix is thick and white, then sperm won't be able to live as long as it would closer to ovulation when the mucus is more of the consistency of egg white. Without the right fertile conditions, the sperm will die within a few hours. So fertilization involves the fusion of 2 haploid gametes. Each of those gametes contain 23 chromosomes, and then they produce a zygote that contains 46 chromosomes, or 23 pairs of chromosomes. So one set of these gametes comes from the sperm, and the other set of gametes comes from the secondary oocyte. So the sperm are delivering the paternal chromosomes to the site of fertilization, and the oocyte delivers the maternal chromosomes and also provides all the cytoplasm, like organelles, like the nutrient pool, like genetic programming, to support the development of the embryo for nearly a week. At fertilization, when it occurs, the diameter of the secondary oocyte is more than twice the entire length of the sperm. So we'll start at the beginning. Ovulation releases a secondary oocyte in metaphase 2. The secondary oocyte is surrounded by a thick glycoprotein layer that's called the zona pellucida. Right outside the zona pellucida is a layer of follicle cells called the corona radiata. The corona radiata is necessary for protection as the secondary oocyte is pushed out of the ovary through the ruptured follicle wall and as it makes its way to the infundibulum in the uterine tube. The oocyte travels a few centimeters into the ampulla toward the isthmus, and that is where the secondary oocyte will be fertilized. If the secondary oocyte is not fertilized, the oocyte then disintegrates without completing meiosis. The spermatozoa travels to the ampulla of the uterine tube. The sperm are motile from the seminal secretions, which caused capacitation and allowed the flagella to move. The sperm then has to be exposed to the conditions in the female tract. The uterine tube cells, called PEG cells, release secretions that help with this capacitation. The exact mechanism is unknown on how this occurs, but the secretions from these uterine tube cells is necessary for the sperm to be fully capacitated. It's also unknown how the sperm finds the oocyte, but they seem to know which uterine tube to swim into. It should take sperm several hours to reach the isthmus, but it only takes 30 minutes to 2 hours because of the contractions of the uterus and its muscular contractions, as well as the current of the fluid in the uterine tube from the movement of the cilia. Of 200 million sperm from one ejaculation, only about 100,000 of them will enter the uterine tube, and fewer than 100 of them will reach the isthmus. A sperm count that's below 20 million per milliliter is considered sterile. Only one sperm is needed to fertilize an oocyte, but dozens are required for successful fertilization to occur. So here we're looking at the secondary oocyte and the first polar body that are being formed from meiosis II. Remember, at the beginning of meiosis II, there was the primary oocyte, and the primary oocyte started to undergo meiosis II, and as it split, one of the daughter cells would become a first polar body, and the other daughter cell would become the secondary oocyte. So here we can see that both the first polar body and the secondary oocyte are wrapped together within the zona pellucida and the surrounding corona radiata cells. We also see the chromosomes of the secondary oocyte are at the metaphase plate, so they're right in the middle of that cell, and so that cell is halted in metaphase 2. During fertilization, the sperm penetrates the corona radiata. Enzymes are released from the acrosome of each of the sperm. The acrosome is in the head of the sperm. The enzyme called hyaluronidase breaks down the bonds between the follicle cells of the corona radiata. It actually takes dozens of sperm to release hyaluronidase before sperm can reach the oocyte. No matter how many sperm slip through the gap, though, that's made by the hyaluronidase, only a single sperm fertilizes the oocyte. That sperm must have an intact acrosome. The sperm binds to sperm receptors in the zona pellucida, and that triggers the rupture of the acrosome, and the chromosomes are released. The hyaluronidase then digests a path through the zona pellucida to the surface of the oocyte. The sperm and the oocyte membranes begin to fuse. The fusion triggers what we call oocyte activation. Oocyte activation is a series of oocyte metabolic activity. The sodium channels on the oocyte open, and sodium moves in, and the oocyte depolarizes. The depolarization causes the release of calcium from the smooth ER into the oocyte cytoplasm. Calcium, then, has the following effects. First of all, it inactivates sperm receptors and hardens the zona pellucida to prevent fertilization by another sperm. So basically it blocks polyspermy, which would be if more than one sperm were to fertilize the same secondary oocyte. If that were to occur, that would create a zygote that's not capable of developing normally. Calcium also triggers the completion of meiosis II. The fertilized oocyte is now called an ovum, and the first polar body forms a second polar body. Lastly, the calcium triggers the messenger RNA in the cytoplasm of the ovum to become activated so that protein synthesis accelerates quickly. After oocyte activation is completed and meiosis is completed, the ovum reorganizes into the female pronucleus, and the male nucleus swells and forms the male pronucleus. DNA synthesis takes place in both the female and the male pronuclei, and those chromosomes duplicate. The male and the female pronuclei move toward each other, and their chromatin condense into chromosomes, each with duplicated chromatids. The centrioles from the male that were donated from the male form the spindle apparatus, and the pronuclei around the female and the male pronuclei dissolve, and the maternal and paternal chromosomes line up in the middle. This combining is called amphimixis. At this point, the cell is now called a zygote. The zygote contains the normal diploid complement of 46 chromosomes, or 23 pairs of chromosomes. At this point, fertilization is complete, and this is known as the moment of conception. At this point, gestation occurs, and gestation consists of 3 stages of prenatal development. There's the first, the second, and the third trimesters. And the very first thing that happens in gestation is that a cleavage occurs where the cell divides to form 2 blastomeres. This occurs 30 hours after fertilization. So that's what we will look at next. Gestation consists of 3 stages of prenatal development. There's the first, the second, and the third trimesters. The first trimester is the period of pre-embryonic, embryonic, and early fetal development. The second trimester is dominated by the development of fetal organs and organ systems. The third trimester is characterized by rapid fetal growth and the deposition of adipose tissue. So let's look at the first trimester. Pregnancies are clinically dated from the last menstrual period that a female has had, or her LMP, which is usually 2 weeks before ovulation and conception. So we know that fertilization is going to occur very close to the time of ovulation, but since it's very difficult on most women to tell exactly when they are ovulating, the pregnancies will be dated by the doctors at their last menstrual period. So from day 1 of their menstrual period, that will be considered day 1 of gestation, even though they actually won't have conception occur for 2 weeks later. There are 4 general processes that we're going to evaluate or look at. 2 of the processes occur in the pre-embryonic period. They're called cleavage and implantation. And then there are 2 processes that occur in the embryonic period, which are called placentation and embryogenesis. Cleavage is the cell division that occurs from fertilization to implantation in the uterus. This is where a zygote becomes a blastocyst and then a preembryo. Implantation is when the blastocyst attaches to the endometrium of the uterus. Then we have the embryonic processes. Placentation is when the placenta forms and the blood vessels form around the blastocyst, and exchange between the maternal and the embryonic circulatory systems occurs. Then in the second process of embryonic development, which is called embryogenesis, a viable embryo will be formed. So let's look a little bit closer at cleavage. Cleavage is a sequence of cell division that begins immediately after fertilization. It produces an increasing number of smaller and smaller daughter cells, and then it ends at implantation in the uterus. During cleavage, the secondary oocyte completes meiosis II and the cells begin to divide. The first cleavage is completed about 30 hours after fertilization. The first cleavage produces a preembryo with identical cells called blastomeres. So now there's two cells that are identical. Subsequent divisions after that take place at 10 to 12 hour intervals. Initially, all the divisions of these cells are going to occur simultaneously. After 3 days of dividing, or of cleavage, the preembryo is a solid ball of cells called a morula. Morula means mulberry, and this solid ball of cells resembles a mulberry. The morula reaches the uterus on day 4 or 5. Over the next 2 days, the morula will become a blastocyst. So let's take a little closer look at this process. So by day 6 and 7, the blastomeres form a blastocyst. The blastocyst has a hollow center called a blastocele. The blastomeres are different sizes and shapes. The outer layer of the cells is called a trophoblast. Tropho means food, so the trophoblast provides nutrients. The trophoblast releases enzymes that dissolve the zona pellucida. The process of dissolving the zona pellucida is called hatching, like the hatching of an egg. A second group of cells cluster at one end inside the blastocele called the inner cell mass. It's this inner cell mass that will become the embryo. Once the blastocyst is hatched, it's exposed to the glycogen-rich fluid of the uterine cavity. The fluid comes from the uterine glands. The blastocyst starts to enlarge. When it's fully formed, the blastocyst contacts the endometrial lining and implantation takes place. Day 8 is when the blastocyst contacts the endometrial lining. At this point, the trophoblast cells divide rapidly and they become several layers thick. The cells closest to the interior of the blastocyst form an inner layer called the cytotrophoblast. Near the endometrial wall, the cytoplasm walls of the trophoblast cells disappear, creating cytoplasm with multiple nuclei. These combined cells are called the syncytiotrophoblast. Syncytiotrophoblast cells secrete hyaluronidase. Hyaluronidase erodes the uterine lining so the blastocyst can dig deeper into the endometrial lining. The maternal cells repair the surface and surround the syncytiotrophoblast. The syncytiotrophoblast continues to enlarge. Erosion of the uterine glands release nutrients for the syncytiotrophoblast. The nutrients diffuse to the inner cell mass. This provides nutrients for the growing embryo. The maternal blood extends into the trophoblast through lacunae. Finger-like villi extend away from the trophoblast and grow until day 21. Syncytioblast spreads and breaks down larger maternal blood vessels to increase blood flow to the lacunae. At this stage, the inner cell mass is unorganized. By implantation, the inner cell mass separates from the trophoblast. This creates the amniotic cavity, which is a fluid-filled chamber. The inner cell mass is 2 layers at this point. There's a superficial layer facing the amniotic cavity and a deeper layer facing the blastocele. Implantation will occur in the fundus or the body of the uterus. An ectopic pregnancy is if implantation occurs elsewhere, for example, in the uterine tubes. Ectopic pregnancies are not that common. 0.6% of all pregnancies are ectopic. An ectopic pregnancy is not viable, and it's actually life-threatening to the mother. The next phase is embryogenesis, or the embryonic period, and this occurs from implantation to week 9. There's a very apparent order to this period. Differentiation involves changes in genetic activity of some cells, but not in others. Differences in the cytoplasm of cells trigger changes in the genetic activity, which leads to further alterations in the cytoplasm. This creates cells with very diverse fates. As development proceeds, the cells release chemicals that affect the differentiation of other cells. This is called induction. It involves a series of interacting steps. During embryogenesis, embryonic structures are formed. Embryogenesis starts at the end of the 2nd week when the inner cell mass is formed, and it ends at the 8th week when the embryo becomes a fetus. Embryogenesis begins with early embryogenesis. During early embryogenesis, germ layers are formed and extra embryonic membranes are formed. At day 10, cells from the superficial layer migrate around the amniotic cavity to form the amnion. The cells from the deeper layer migrate around the outer edges of the blastocele and begin to form the yolk sac. The yolk sac is the primary nutrient source for the inner cell mass. By day 15, a 3rd layer of cells forms between the superficial and deep layers of the inner cell mass. The process of cells migrating from the superficial cells toward the central line, which is called the primitive streak, is called gastrulation. Gastrulation creates 3 distinct layers: the ectoderm, the mesoderm, and the endoderm. The ectoderm is the superficial cells that didn't migrate. The mesoderm is a poorly organized middle layer, and the endoderm is the layer that faces the yolk sac. These layers are also called germ layers. They form an oval 3-layered sheet called the embryonic disc, and this is what will form the body of the embryo. Each of the germ layers will produce specific structures of the embryo. The ectoderm forms the epidermis, glands like the sweat glands and mammary glands, the brain, the spinal cord, the pituitary gland, and the adrenal medulla. The mesoderm forms structures like the dermis, the hypodermis, the skeleton, the muscles, the adrenal cortex, the cardiovascular structures, lymphatic structures, kidneys, gonads, and serous membranes. The endoderm forms the thymus, the thyroid, the pancreas, the urinary bladder, gamete stem cells, and distal reproductive ducts. All other cells become the extraembryonic membranes. The extraembryonic membranes are the yolk sac, the amnion, the allantois, and the chorion. The yolk sac is the primary nutrient source for the early embryo. It will become the embryonic blood vessels in the umbilical cord. The amnion is the ecdermal cells that line the amniotic cavity. The chorion are the mesoderm cells that spread around the entire blastocyst. Its role is to exchange nutrients, gases, and wastes between the embryo and the mother as the distance between the embryo and the trophoblast increase. At this point, blood vessels develop in the chorion. The appearance of blood vessels is the first step in the formation of the placenta. As embryogenesis takes place and the body of the embryo and its internal organs begin to form, we will see the embryonic disc produce a bulge called the head fold and a tail fold. So here again we see at week 2, we can see the 4 extra embryonic membranes. The yolk sac is the primary source, again, for the early embryo. It will become the embryonic blood vessels in the umbilical cord. The amnion are the ectodermal cells that line the amniotic cavity. The chorion are mesoderm cells that spread out around the entire blastocyst. Its role is to exchange nutrients, gases, and wastes between the embryo and the mother as the distance between the embryo and the trophoblast increase. The allantois becomes the connection between the fetal bladder and the yolk sac. Here you can see a drawing of the embryo and its position with the head fold and the tail fold. We can see the chorionic villi. The chorionic villi will help to exchange nutrients, gases, and wastes between the mother and the embryo. Blood flow through the embryonic blood vessels begins early in the 3rd week, and the embryonic heart also begins to beat. The blood supply to the chorionic villi is from the arteries and veins of the allantois. At first, the entire blastocyst is surrounded by chorionic villi until week 4. Then the embryo, amnion, and yolk sac are suspended in a fluid-filled chamber. A body stalk connects the embryo and the chorion, and this umbilical stalk contains both the allantois and the blood vessels. The yolk stalk connects the embryo and the yolk sac. Chorionic villi enlarge and more maternal blood vessels are eroded to supply the lacunae with blood. During week 5, the placenta is formed. Placentation is the formation of the placenta. The placenta is formed from both embryonic and maternal tissues. The placenta will be fully formed by the end of the 3rd month. During placentation, blood vessels appear in the chorion. Here you see the chorion, which are mesoderm cells spreading around the entire blastocyst and forming chorionic villi. The allantois are the endoderm cells. The base will become the urinary bladder. The yolk sac is the primary nutrient source for early embryonic development and will become blood vessels. And the amnion are ectodermal cells lining the amniotic cavity. The embryonic period has ended at the end of the 10th week of pregnancy, and the embryo is now considered a fetus. A fetus is a developing baby beginning in the 11th week of pregnancy. We can see here how at the end of the 1st trimester, the fetus has moved farther from the placenta, but remains connected to the umbilical cord, and the umbilical cord contains the allantois, the placental blood vessels, and the yolk stalk. Blood flows from the fetus to the placenta through paired umbilical arteries and then returns in a single umbilical vein. Gases, nutrients, and wastes are exchanged between the fetal and maternal bloodstreams. The fetal hemoglobin has a higher affinity for oxygen than the maternal hemoglobin does, so oxygen diffuses into the embryonic blood. There is no mixing of maternal and fetal blood that takes place because layers of the trophoblast separate the two. The maternal blood flow to the placenta is extensive. The placenta places a considerable demand on the maternal cardiovascular system. If torn, it can be fatal to both the fetus and the mother because massive hemorrhaging could occur. The 1st trimester ends at the end of the first 12 weeks. Events in the 1st trimester establish the basis for the next phases of organogenesis, which is organ formation. The rudiments of all major organ systems are formed by the end of the 1st trimester. The 2nd trimester of pregnancy is from week 13 to week 28. It's roughly months 4, 5, and 6. In the second trimester, the fetus grows to 1.32 pounds. The fetus during the second trimester grows faster than the placenta. The mesoderm from the amnion and chorion fuse to form the amniochorionic membrane. In the third trimester, most organ systems are ready for normal functioning without maternal assistance. The rate of growth slows. The weight gain in the third trimester is greater than the first and second trimester. In the last 3 months, the fetus gains 5.7 pounds. The average full-term weight is 7.05 pounds. This ends our first video on development. There will be another video on development where we talk about hormones and we talk about changes in the maternal system. We'll talk about abortion and childbirth. So I'll see you next time.